Hello, everyone. It is my pleasure to introduce our recent research to you. Whole-exome sequencing reveals an Fcgbp variant associated with spontaneous intraabdominal hemorrhage in severe acute pancreatitis. Acute pancreatitis is an acute inflammatory condition of the pancreas. Approximately 20% of acute pancreatitis cases progress to severe forms. Spontaneous intraabdominal hemorrhage is particularly lethal, with a mortality rate as high as 54.2%. Our study aims to explore the genetic regulatory background of SIH and SAP. We identified a strong association between mutation in the Fcgbp gene and SIH. Further in vivo and in vitro experiments demonstrated that Fcgbp expression levels are closely linked to the bleeding risk. We retrospectively reviewed all acute pancreatitis patients admitted to our center, and 49 cases were diagnosed as SAP, complicated by SIH. We selected 45 SAP patients without SIH to establish a primary control group. Additionally, we leveraged a prospective registry-based cohort from our center, comprising 510 acute pancreatitis patients as an independent secondary control group. Systematic analysis of the whole-exome sequencing data from these three groups yielded a series of significant findings. To identify the mutations most likely associated with SIH, we conducted rigorous quality control and filtering of all mutations. This process ultimately identified 1,322 candidate mutation sites. Figures B and C and D illustrate the principal component analysis results, their chromosomal distribution, and detailed information on mutation types, sample distribution, and gene distribution. This study is the first to reveal the genetic mutation characteristics associated with SIH and SAP. Furthermore, we performed a variety of comparative analyses of mutation distributions between the case and control groups. We identified a frameshift deletion mutation in the Fcgbp gene on chromosome 19, which showed a strong association with spontaneous intraabdominal hemorrhage. This mutation was highly prevalent in the SIH patient group. We randomly selected acute pancreatitis patients carrying the Fcgbp mutation and non-carriers. Serum Fcgbp protein levels were measured, revealing significantly lower levels in mutation carriers. We further performed animal experiments. Fcgbp-knockdown mice were subjected to an SAP model, which exhibited significantly worsened pancreatic and pulmonary injuries. Importantly, red blood cell leakage was visibly detected in the pancreatic interstitial spaces of Fcgbp knockdown mice. These findings provide compelling evidence that Fcgbp deficiency is associated with vascular injury and bleeding. To further investigate the protective role of Fcgbp in preventing SIH, immunofluorescence analysis on mouse vascular tissues revealed a high degree of colocalization between Fcgbp and alpha-SMA, suggesting that Fcgbp within the vascular wall may originate from fibroblasts. We cultured human vascular-derived fibroblasts in vitro and performed transcriptomic sequencing after knocking down Fcgbp. Fcgbp-deficient fibroblasts exhibited 309 downregulated genes, which were significantly enriched in pathways related to extracellular matrix, extracellular vesicle structure, and collagen-containing extracellular matrix. These findings indicate that fibroblast-derived Fcgbp within the vascular wall plays a vital role in maintaining vascular stability. A reduction in Fcgbp expression due to mutations may compromise vascular integrity, thereby increasing the risk of hemorrhage. Finally, we evaluated the clinical significance of the identified Fcgbp mutation. Clinically, acute pancreatitis patients carrying the mutation exhibited significantly higher mortality rates and increased incidences of acute kidney injury. To assess the mutation's predictive value, we developed multiple outcome prediction models for acute pancreatitis. Including the mutation as a variable significantly improved the model's predictive performance. These findings suggest that the Fcgbp mutation could serve as an effective predictive marker, enhancing early diagnosis and risk assessment for complications and prognosis in acute pancreatitis. In this study, we provided the first report on the genetic mutation characteristics of patients with SAP, complicated by SIH. Through systematic analysis and experiments, we identified a significant association between Fcgbp gene mutation and the risk of SIH. These findings provide critical insights into the mechanisms underlying SIH and lay the foundation for developing effective therapeutic strategies.